Hello all. I received an email from the American Pharmacists Association last week in regards to a paper that was retracted from both the Lancet Journal and the New England Journal of Medicine. It had to do with hydroxychloroquine, and I think it is a good example of why journal clubs are important. There were over 100 physicians that wrote in that caused this article to be retracted, and it shows why these things are important. So I wanted to go over the details of this study and then ultimately why it was retracted. It was an observational study using data collected from a multinational registry from 671 hospitals across 6 continents. The title is Hydroxychloroquine or chloroquine with or without a macrolide for treatment of COVID-19, a multinational registry analysis. And like I said, it's an observational study, so it's obviously not the gold standard. To be included in the study, patients needed one positive test result either from a high-throughput sequencing or reverse transcription quantitative PCR assay of nasal or pharyngeal swab specimens. I looked into both of those tests and they are approved as diagnostic tools. There's some issues with the specificity of the reverse transcriptase assay. Um, it might be more apt at catching more developed cases rather than cases in the first week, but I'll include the links to those studies in the description as well. People to be included also needed to be treated with hydroxychloroquine, chloroquine, or hydroxychloroquine with a macrolide or chloroquine with a macrolide within 48 hours of a positive test result. The patients who were excluded were either on other medications, started one of those treatments while on mechanical ventilation, or they received a chloroquine analog more than 48 hours after a COVID diagnosis. So altogether, they included 96,032 patients. Those are the number of people to take part in the study. That's a remarkable sample group. And like I said, it took place across 6 continents. So you can expect to collect that type of data from a pandemic. It is still a very large sampling. So 1,868 people received chloroquine, 3,783 received chloroquine with a macrolide, 3,016 received hydrochloroquine, and 6,221 received hydrochloroquine with a macrolide. And if you don't know what a macrolide is, macrolide is antibiotic or antifungal. Oftentimes it's substituted for a penicillin if the patient's allergic to penicillin, and it inhibits protein synthesis in those bacteria. Something to note is they may cause arrhythmias, specifically QT elongation. So this study looks at both mortality in the treatment groups and also de-novo ventricular arrhythmia. Those are the two dependent variables that we're looking for. So out of those people, those were designated to the treatment group, and then the rest of the 81,144 went to the control, where they did not receive treatment. The confounding factors that this study aimed to negate were age, sex, race, ethnicity, body mass index, underlying cardiovascular disease, and other risk factors such as lung disease, smoking habits, other comorbidities, as well as disease severity. Now, they used 2 measures to decide on the level of disease severity. They used oxygen levels, and they also used this test called the sepsis-related organ failure assessment, also known as the QSOFA. QSOFA relies on three measures, and points are allotted for low blood pressure, high respiratory rate, and change in mental status. The higher your score, the greater of a likelihood of a poor outcome in the ICU. So high score bad, low score good. It's out of 3 possible points. However, there was a article published in Springer that challenges the utility in measuring the severity of COVID-19. So the sepsis-related organ failure assessment is called into question, and its ability to rank severity of COVID-19. So it's one of those things that could be improved upon. And to analyze the data, a Cox proportional hazards regression analysis was used to visualize the associated risk of mortality and developing arrhythmia as they relate to the 4 treatments. On top of that, propensity score matching was then used to reduce the bias resulting from confounding variables. And then tipping point analysis was also used, to get a read on the unobserved confounding variables. Excuse me. The results, I don't want to talk about the results too much because this article was retracted, but they did find that there was increased arrhythmias with hydroxychloroquine and chloroquine analogs with macrolides in combination treatment. They found that the use of regimen containing hydroxychloroquine or chloroquine with or without a macrolide was associated with no evidence of benefit, but instead was associated with an increase in the risk of ventricular arrhythmias and a greater hazard for in-hospital deaths with COVID-19. So, greater risk of mortality, greater risk of arrhythmia. Two really it's unexpected results from what was a promising treatment. So these unexpected results obviously raised some red flags, which brought in some critiques. Like I said, over 100 physicians signed an open letter. They wanted more data. So here are the reasons for why this article was retracted. The official statement was the article was retracted on the grounds of the inability to confirm the veracity of the data supplied by SugarSphere Corp. Now, SugarSphere Corporation is the company responsible for collecting the data. Their methods of doing that weren't transparent, and they were corrupt. It wasn't done correctly. Some other suspicious things about this paper. There's only 4 authors on a paper dealing with such a large sample size, with 96,000 plus people participating. It's typical that you would have hundreds of authors working on it, but that is also something of concern. More so, the Australian data represented in this study did not match the Australian government reports. According to this study, there were more in-hospital deaths than deaths throughout the entire country. And that didn't make sense because surely there are more people dying at home, and those numbers they don't match up. I came across this odd table. If you look into this study, you might come across table S3, where different confounding variables across continents were extraordinarily consistent to the point where it looks doctored. That was the major thing that stuck out to me. For instance, smoking prevalence across the 6 continents ranged from 9.4% to 10.3%. And all the data seemed to be within 1 or 2 percentage points in a range. So that didn't really make a lot of sense to me. However, Dr. Steinhardt created a spreadsheet, and he figured out that there was some statistical processing that had been done to the numbers, and then those numbers had been presented as raw data rather than processed. So no foul play expected, but it is odd that that data was reported as raw. The authors did issue an apology, saying, "We all entered this collaboration to contribute in good faith and at a time of great need during the COVID-19 pandemic. We deeply apologize to you, the editors, and the journal readership for any embarrassment or inconvenience that this may have caused." And unfortunately, it did cause an inconvenience. The World Health Organization suspended all trials on hydroxychloroquine because of this paper. Obviously, safety first is kind of the motto moving forward, and this article alone put that into question. So it did have impact, but because over 100 physicians were able to interpret and critique the article, they were able to have it retracted, and trials were able to continue. So I think it's a good thing we're doing. Thank you.